All right, so welcome to this edition of Root Causing Health podcast. This is going to be a little bit of a different format than what we've done before. We're going to be a little bit more conversational. Basically, there's this new paper that came out. It's about 11 pages of paper and 12 pages of conflicts of interest section with all the pharma funding, but it's from the EAS, and it's another consensus statement. So, as Nathan will tell you, that's not really science.
Yeah, that's not how science is done. And this one is titled Low-Density Lipoproteins Cause Atherosclerotic Cardiovascular Disease, Pathophysiological, Genetic, and Therapeutic Insights, a consensus statement from the European Atherosclerosis Society panel,
Yes.
which is just riveting.
Yeah, so basically I think the way this works is that every few months they get concerned that people have forgotten that there's a consensus, so they release another paper to talk about it ad nauseum. And so we just wanted to, I think a lot of people were curious like how we read papers, because it can be a little bit intimidating to people who are just starting out or haven't read a lot before. So I figured we would just sit down and explain to you our thought process as we go through this paper. Because I think to a lot of people, they'll see this and they'll say, well, fuck, like it's true. You know, there are a lot of people with names and asterisks and funding have said it to be so it must be true. I think a more direct reading of the paper and explaining bit by bit how well they make their case and an assessment from our perspective will help people, temper their fears along those lines.
And I think that there's a lot that can be broken down in this paper, and if you really read into it, just like some of the previous papers, really it raises more questions than it provides answers.
I guess we can start at the top with their introduction. They go over, they say again, atherosclerosis starts early, even in childhood. They always cite that. I forget which paper did they actually cite for that. Was it the Bogulosa Heart Study or? Yeah, Bogulosa Heart Study, 1998. Interesting. So one of the interesting things about these papers is that partially what they do cite and also what they don't cite. So there's not a whole lot of pathology being cited in this paper. There are a couple examples. Most of them are from the Bogulosa Heart Study. Konstantin Velikin, I do not believe is cited, nor is Stebbins, nor are most of the other pathologists that were very popular in this field, which I think is just a little bit interesting. They have a lot of imaging studies, a lot of stuff like that, but they don't really cite pathology with a couple limited exceptions. I don't know exactly why they've chosen the Bogulosa Heart Study. How do you say Bogulosa?
I have no idea.
Okay, we're going with that. Heart Study as their chosen pathology work, but it is not the only one, but they studied they cited a couple times. Most of the research that they'll cite in here is mouse model studies and imaging studies, basically. That's kind of the extent that they'll cite. They don't cite a lot of actual study of actual tissue samples on human arteries, which is kind of interesting.
I think just as an opening line, this is an interesting one, that ASCVD starts early, even in childhood. And that's the only thing that they open up with.
They're trying to get at this idea that atherosclerosis is a slow, chronic, progressive disease, which I think a lot of people, it's not necessarily as obvious as people. I don't know that that's necessarily correct, but...
I also have some notes. Their next sentence is that the non-invasive imaging study revealed that 71% and 43% of middle-aged men and women, respectively, have evidence of, quote, subclinical atherosclerosis. And my note there is, what the heck is subclinical atherosclerosis, and how are we measuring that?
I think it's usually some amount of stenosis as measured by angiographic study or something like that. So they basically say here that extensive evidence from epidemiologic, genetic, and clinical intervention studies has indisputably shown that low-density lipoprotein is causal in this process, as summarized in the first consensus statement on the topic. I found that statement to be pretty interesting because it doesn't, like, the conventional diet-hard hypothesis is saturated fat raises cholesterol, clogs arteries, right? But they didn't say that elevated low-density lipoprotein causes cholesterol. They just said, like, the existence of the molecule, which is a little bit interesting. One of the other things they mentioned in this paper was that the apoB 100 is one of the largest mammalian proteins, which I think is kind of an interesting thing to discuss. Like, I guess there was some great selective pressure to create this garbage particle that, like, it just seemed a little bit interesting to me.
Yep.
So it says, although LDL is unequivocally recognized as the principal driving force in the development of ASCVD and its major clinical sequelae, great word, evidence for the causal role of other apolipoprotein containing apolipoprotein B, apoB containing lipoproteins in ASCVD is emerging. So I guess they're saying that's not only LDL, it's also the couple other...
Any apoB containing lipoprotein.
So VLDL and IDL, of which there were far fewer.
But I also thought it was interesting that it's now, they've kind of flip-flop a little and moved from it's causal in the process to it's a principal driving force. And I don't know that those quite mean the same thing.
Yeah, they've definitely abused the word cause. I think typically cause is held to be a sine qua non, which means that, like, I mean, I guess they say it's cause because it's always there. Like, you always have genes for apoB100 with limited exceptions. Therefore, like, that, the fact that you have a gene for apoB is a cause of heart disease, which is kind of nonsensical to me. Oliver's saying hi. He's a little bit bananas right now. What do you want, dog? Hello. Yes. That, to me, like, that's just a problematic thing to say because apoB...
We're not going to live without it.
Yeah, it's like saying, like, glucose is causal. I think Ted Naiman had the graphic on this where, like, gravity is a cause of plane crashes. It's the same kind of, like, idea that's getting at, which I think is probably a pretty accurate characterization of why that statement is problematic. Like, a sine qua non is typically, like, a cause is typically held to be a sine qua non, which means that if the thing does not exist, there will not be disease. And the problem is that people with low LDL get disease. Therefore, how can... but again, this whole problem is there isn't... It's very difficult to tell what the hypothesis is when you're reading this research because that statement is very vague. It just says that LDL is causal in this process. Like, just what about it? Can you give me any more information? Is it the size, the quantity? And then they just kind of then go and they, like, word vomit for like 15 pages on, like, just different random facts about LDL and its associations and things. And that's one of the first thing, like, right off the bat that makes me kind of angry. It's why I brought this wine to this evening to help me get through this paper. It's not as bad as some of the papers I've read. But anyways.
Yeah, some nice Bordeaux. There you go. Or maybe it's shitty Bordeaux. I don't really know.
No, this is a nice Bordeaux.
Oh,
Came from France, actually. So that's
Perfect.
how you know it's good. It's actually pretty dry, too, and I got it from the convenience store down the road. Anyways.
Very good wine, then.
$15. All right.
So should we jump straight to the circular causal inflammatory
Let me see.
story here?
So, well, I mean,
I like that they open with, we don't understand it.
Yeah,
fully.
so then they say the third paragraph starts with, the pathophysiological and genetic components of ASCVD are not fully understood, full stop. And I'm like, yes, I agree with you, good job. We're on the same page.
And we keep going. We have incomplete understanding, for example, of the factors controlling the intimal penetration and retention of LDL. And then they move on to the circular hypothesis that we love so much, the subsequent immunoinflammatory response of the arterial wall to the deposition and modification of LDL.
Yeah, so their kind of root cause hypothesis is that, like, LDL exists, and then that causes macrophages to be activated, which then causes more LDL to be oxidized, which then causes more immune activation. They actually had a really funny statement where they actually very much said it was a circular hypothesis. And I like call this out a couple times because they
it also wasn't clear to me in a few places. They said, like, oh, the macrophages cause more ox-LDL, and that was there was no cited source on that. This
is one of my favorite quotes. They go, thus retention and subsequent modification of LDL elicits both innate and adaptive cellular and humoral immune responses that drive inflammation in the artery wall. No citation provided. Then they said, disrupting this vicious cycle by targeting inducers and mediators may provide alternative approaches to halting atherogenesis at specific stages. Again, as I described, this is kind of like the canonical sand in the gears way of thinking about atherogenesis. I don't like that, needless to say. Let's see. Do you have anything? I guess we can just go through and call out things as we...
Yeah, I mean,
interesting.
Yeah, I think the next bit that they went to was transcytosis, which was covered, which is how lipoproteins are moved across the cell into the arterial wall. And they had an interesting one here. They said that despite the relevance of endothelial transport during atherogenesis, the molecular mechanisms controlling the process are not fully understood. Although they have said that a considerable body of evidence in recent years has challenged the concept that movement of LDL occurs through passive filtration, i.e. as a function of particle size and concentration, across a compromised endothelium of high permeability. So that theory has apparently been dropped.
That's really funny because that's why they don't say a high concentration of LDL causes this problem, because that statement contradicts that idea, right? Because as soon as all of a sudden you get down a rabbit hole of like, okay, well, if it's active transport, what are the factors governing the active transport? How dependent is the rate of transport on the hypercholesterolemia or whatever? The other interesting thing... So they went over a number of things on this paper, one of which was they were talking about the fact that in the arterial region that the transcytosis was somewhat independent of the LDL receptor, and they talked about DOC4 and SRB1. Mm-hmm. And one of the things that I did, of course, is every time I see one of these things, I Google the thing followed by endotoxemia just to see like what pops up. And sure enough, there's a lot of discussion on like sepsis and SRB1 and endotoxemia, which I thought was kind of interesting.
That was actually, I had that highlighted as well. And I was thinking, hey, these two receptors, SRB1 and DOC4, are expressed in more quantity in atherosclerotic arteries. So like what's actually driving that? It's probably the inflammatory response, not the LDL itself.
And again, they're getting at this kind of
because
idea.
These receptors are responsible for pulling in more LDL. So if we think that the body is trying to cause this, why would it express more of these things?
That's actually a recurring theme that I highlighted multiple times. They say a lot of statements in here that basically directly oppose their hypothesis. They talk a lot about like mutations in... one of the things they mentioned, I think it was the next page, was talking about how GWAS... some of the higher GWAS associated genes. So when they do a gene-wide associational study in atherogenesis, arterial wall proteins are strongly associated with atherogenesis. I'm like, again, like there are so many of these comments they make that are not at all arguing their.
Yep. And related to that, I have another one in here. Transcytosis of LDL across endothelial cells can be increased by activation of the NLRP3 inflammasome, or by hyperglycemia. So.
A lot of things.
That's NLRP3 inflammasome can be activated by endotoxins, I'm sure, and other nasty stuff in the blood, lead or what have you. And hyperglycemia, we know that's no good. So that can help explain why you'd see increased atherosclerosis and diabetes as one potential option.
Lots of pluricausality. But then on page 3 they go, this was one of the more interesting statements. The mechanisms that underlie increased rates of LDL transcytosis during hypercholesterolemia remain unclear. In other words, they don't know why more cholesterol moves into the artery wall when people have high cholesterol. I think that gets at the very core of like, what the hell is this LDL hypothesis? Like the conventional implication has always been that it's a concentration gradient defined problem, basically, and that statement kind of shoots that in the foot, because it's essentially admitting, maybe that's a little bit of a stretch, but I think it's essentially admitting that hypercholesterolemia per se does not cause increased transcytosis, which might mean that hypercholesterolemia and increased transcytosis are like associated, not mutually causal. Like there are a lot of other models that could explain why that's going on, but I just thought that was kind of interesting.
Yeah.
This next section.
So now we talk about retention.
Mm-hmm.
And I think, this is selective retention. So that's interesting. So why is it being selectively retained? And this is what I highlighted in the apoB 100 or apoB Triumph of Simplicity paper, that the selective retention is super interesting. And it seems like from our research, endotoxemia can modify, and inflammation can modify the extracellular matrix proteins and increase their affinity for apoB 100. The
proteoglycans appear to be somewhat coupled to the immune response in some fashion in ways that are not entirely understood. We talked about that in our Foundations 3 episode, which hopefully we might publish before we end up publishing this.
Maybe.
Who knows? All right. They talked about, again, selective retention. So anytime they're talking a lot about retention, a retention-based theory of atherogenesis is not per se a LDL concentration is causal theory of atherogenesis. They're like, to some extent they are incompatible. If retention is the governing force, then why do we care about the amount of LDL particles? Like,
that other paper called out that it wasn't a function of how much LDL there was.
Yeah, so this is one of the few pathology studies they cite, but they say autopsies studies in young individuals demonstrated that atherosclerosis prone arteries develop intimal hyperplasia, a thickening of the intimal layer due to the accumulation of smooth muscle cells and proteoglycans. And I was like, yes.
Yep.
So they're...
True facts.
Yes, I agree. In contrast, atherosclerosis-resistant arteries form minimal to no intimal hyperplasia, would suggest that intimal hyperplasia might be the initiating factor in atherogenesis, or at least one of the earlier steps.
Yep.
Before LDL accumulates again, which is kind of, it feels like they're like continue to throw us bones as we go along. But.
And then they go on to talk about laminar flow and perturbing that and causing matrix protein or matrix proliferation and then subsequent lipoprotein retention. So it seems like the matrix proliferation comes first.
Yes.
Followed by lipoprotein.
Again, our kind of thesis about this paper is that it really doesn't argue their cause particularly well. Like the actual paper does not sufficiently or even vaguely support the hypothesis. Again, this is where the GWAS quote said, is a number of genetic variants strongly associated with ASCVD in genome-wide associational studies occur in genes that encode arterial wall proteins, which either regulate susceptibility to LDL retention or the arterial response to LDL accumulation. I think there are other possible ways in which those arterial wall proteins could mediate them.
Yep.
They tend to not cite things. There are like key places where like.
But I do like that they mentioned hemodynamics and shear stress.
Yeah.
And that's important stuff.
Kudos to them.
That does not get mentioned very often. So I'll give them some credit for that.
They talk about apoB 100.
Did you know that they're pseudomyocellular, quasi-spherical, and plurimolecular complexes?
I did not.
Well, there you go.
There's
True
a lot of
facts
vernacular
about
that they whip
LDL
out during these things. Spectrum of different particle subclasses with different physiochemical probabilities.
I don't know why they're talking about all this, to be honest. I thought they just said that particle size didn't matter, and now they keep yammering about
it's filler.
Classes
it's like a credential building type thing, in my opinion.
There's 3 types: large and buoyant. I don't know why. How many different, large, buoyant, huge,
they
big.
do actually. They go along and they talk about the association between particle size, and then they talk about counterexamples between the particle distribution and the association with ASCVD. Like one of the comments they called it was that familial hypercholesterolemia associates with a quote unquote favorable lipid profile. I think in terms of like small dense LDL ratio, if I remember correctly. Yeah. However, this pattern is also seen with familial hypercholesterolemia in which LDL levels are high. They're like, I don't know how to explain that.
I did think it was pretty interesting that they have the 3 classes with low plasma triglycerides, average, and high plasma triglycerides. And I'm pretty sure that would correlate pretty well with insulin.
Oh, yeah.
Low levels of insulin and diet, I assume.
I tweeted out, I was like, I didn't know they were in favor of the LMHR triad. Like,
yeah, they basically are.
Because they talk a lot about like the association between, they're like, what did they say? One of the comments was that when triglyceride is very low, that LDL can actually be secreted from the liver directly or something like that, or IDL particles. The liver can produce a range of particles varying in size from large VLDL1, medium-sized VLDL2 to LDL depending upon hepatic triglyceride availability. It's kind of interesting. The rate of VLDL production is also... Oh my God, dog, what are you doing? The rate of VLDL production is also influenced by metabolic factors such as insulin resistance.
Well, there you go.
There we go. Where is it? Individuals with plasma triglyceride in the normal range release VLDL1 and VLDL2 from the liver, which are delipidated rapidly to IDL and then principally to LDL of medium size. Thus, the LDL profile is dominated by LDL2. In contrast, people with low plasma triglyceride levels of 75 milligrams per deciliter or lower have highly active lipolysis and generally low hepatic triglyceride content. Consequently, hepatic VLDL tend to be smaller, and indeed, some IDL or LDL size particles are directly released from the liver. The LDL profile displays a higher proportion of larger LDL1 and is associated with a healthy state, as in young women.
Well, gosh, Nick, do you think that might explain the lean mass hyperresponder LDL phenotype?
Do we think that might explain the association of all of this stuff?
Pretty sure both of us have no hepatic triglycerides, or as a healthily small number, since we were MRI'd and we didn't have a fatty liver.
Yeah.
So I'm pretty sure that could explain it. They've answered their own question
I know.
here.
They're like, well, we're done now. We can move on, and we can move on. All right. I thought that was interesting. But then they talked about FH. I think my personal problem with lipids are that it is just there is a lot of noise and a lot of input variables. So you got to denoise it. I think the signal's just noisy enough that it can be...
It's a big swamp. We could spend a lot of time playing with particle sizes and tracking them around. And
I just think that like slicing and dicing it infinite different ways is not going to really get us to where we need to be.
We've been doing that for a long time, and we still have a lot of CVDs.
I think there's a better thing to measure. So anyways, now we get into some of the more arguments about how LDL is causal. So they said formation of macrophage-derived foam cells. Yeah, I was worried that foam cells hadn't been mentioned in about 2 paragraphs. So
here we are.
Foam cells are back. So uptake of aggregated LDL particles. They say aggregation may occur by non-enzymatic or enzymatically induced mechanisms. When you see may in one of these papers, it means may or may not. So they say aggregation may or may not occur by non-enzymatic or enzymatically induced mechanisms. Like, it's not even a statement. It's like, are there other options? Like non-enzymatic or enzymatic? It's like all possible mechanisms.
All. Yeah, that's all possible.
I think that A or not A, like, I don't, like, how much are we paying these people? But oxidation of lipids can occur enzymatically or non-enzymatically. Okay, cool. Release of bioactive pro-inflammatory lipids.
By the way, if it's happening enzymatically, aren't those enzymes there to do something? Aren't they doing that like on purpose or something?
You'd think so.
Yeah. I don't know what myeloperoxidase's job is with regards to apoB 100, but I know that that's a sign of arterial wall inflammation, but I have to look into that one.
They whip out cholesterol crystals. They say that a lot. I don't really know.
I couldn't find a paper on how exactly they're formed. I know that this is like that's like a triple point thing, right? With chemistry, like to get a crystal, you need pressure, temperature, all that kind of stuff. Usually they don't just form kind of willy-nilly, but I don't really know.
Let's see. So one of the ways that they propose that they're causal is induction of an innate immune response involving damage-associated molecular patterns. I find this argument to be stupid. Like, I do not believe the entire purpose of our immune system. It has one purpose in its life, is to differentiate between exogenous and endogenous compounds.
So damps are a thing. You can have sterile inflammation.
Yes, but
I expect that this does become a problem at some point in atherosclerosis when you have tissue necrosis and apoptosis going on in later stages. Then that probably would result in some amount of
this
damp
specifically
release.
says induction of an innate immune response.
Yeah, I mean, I think that seems unlikely.
I just like what they're trying to argue is that at the very tip of the iceberg, the LDL particle wanders in, and then because of its horrifying nature, triggers a damp. I don't buy that model.
Yeah, I don't think so.
I think that all of the research supporting that comes from mouse models. It comes from in vitro studies of pouring shit over cells. I don't think that case has been made. Again, like there's no citations in this paragraph. Convenient. And then they go on and then they'll say that kind of bold statement, and they won't substantiate it with a citation, and then they'll move on to talk about what damage-associated molecular patterns promote recruitment of immunoinflammatory cells. Like, yes, correct. And then leading to locally and potentially chronic inflammation. Again, chronic inflammation, my favorite bullshitism, that can or cannot induce cell death by apoptosis or necrosis, thereby contributing to necrotic core formation. So, like, it is plausible that...
I mean, it seems unlikely that the recruitment of these inflammatory cells would lead to apoptosis and necrosis. That's not really what's supposed to go down there. I guess it could, but
in theory,
that doesn't seem like the most plausible outcome.
And they would say induction of an adaptive immune response.
Oh, we've got both systems involved now.
Subsequent to covalent modification of apoB. Like, and they
just
talk
the
about
aldehydes. Do you know when the aldehydes get in there and they just fuck up your apoB?
Like, I don't... So this...
Or it could be by degradation with the activation of antigen-specific T-cell responses and antibodies.
Possibly.
I love it when my immune system decides to attack all quintillions of my apoB particles.
Like, So, and the other thing is, like, in general, I think it's going to be very difficult in a research environment, no matter where you are, to decouple the modification of these things with metabolic dysfunction. Those two things are probably very coupled. And so I think it's very difficult to differentiate if the causal vector is going through metabolic dysfunction or if the causal vector is going through some modified thing that is substantially a result of metabolic dysfunction. Like, I think that's going to be very fuzzy and very difficult to tease apart. And they always assume, always, that the causal vector is basically like from the modified thing, that the modified thing is the beginning, the root of that tree, which just, I don't know, it happened probably as a feedback loop from previous modified things and macrophages or foam cells. Obviously, foam cells, got to use that word. That, again, bothers me. And I simply don't think that there's very little citations throughout this. They do have 5 on here.
Why did they put 4 on that? I don't know. Weird spot to put. Maybe is that for all of them?
Sometimes I feel like they were just like getting pretty apathetic when they were finishing this paper and just cited 4 things in a row. And it's funny, a lot of times when you go to these citations, like the paper, I have found, I haven't dug through the citation tree enough. There are a couple that I've looked at, but a lot of times in these papers you will find citations that are not actually substantiating the thing they're listed next to, which is, that's why I will usually, especially if there's something that I find that's controversial, I'll go through and actually read that paper. And a lot of times in this paper, a couple of times, things seem to be taken a little bit out of context, or the context from even the abstract was not sufficiently preserved in the, like, it'll be a mouse model study and they won't say in mice. They tend to just say I think we're coming up on that in short order, but a lot of times they will say atherosclerosis and they will not differentiate the fact that they're talking about experimental
versus
they're talking about a mouse aorta
humans.
In an apoE minus mouse, which is experimental atherosclerosis, which looks 0% like human atherosclerosis. It's a fatty streak type thing that you'd expect in FH, and it's not consistent with like the same pathology or same progression. And they'll use those terms interchangeably. And that's where this whole idea that the total lack of a definition of atherosclerosis really starts to bite us, because we just use that word everywhere without regard to like what it means or what we expect to see.
Yep. And moving on, we got a mention of Lp(a), which, in case you didn't know, is composed of an apoA covalently linked to the apoB of LDL. And they mentioned that it's a major carrier of pro-inflammatory oxidized phospholipids. So that's true, I believe, and interesting to note, because I believe Siobhan has talked about this, Siobhan Huggins, about how apoA may be responsible for clearing up oxidized lipids.
Yeah, I think one of the major hypotheses that I would want to rule out is the possibility that a lot of the things we're seeing. So the way I view this paper is that a lot of these things are like, they're looking at it like one level below the surface. They don't quite see the full story, and so they're looking at mechanisms in isolation, and it's why it seems very ADD like. It's just they're talking about a lot of different things because they don't quite see how it fits together. So this hypothesis that's been tossed around that these are disposal mechanisms, I think, is an interesting one to explore, because it could potentially explain why receptors have different affinity for modified LDL, or why these Lp(a) particles always have these oxidized things attached to them.
Also, writing a paper by committee is bound to not end well, and that's another reason that we get kind of crappy papers.
Yeah, I don't know. Anyways, let's see. I had some things highlighted in the next paragraph. Under defined cardiometabolic conditions, a specific LDL subclass may become more prominent as the driver of atherogenesis. What?
I don't know what to make of that.
I don't even know what that's saying. Several biological properties of small, dense LDL could confer height. Like, that doesn't even, they're like, oh, we have a box 2 explaining all the properties which could in vitro
prolonged residence time, increased affinity.
Oh my God.
Enhanced arterial wall penetration.
Okay.
But we just ruled that out. They literally just said that it doesn't just go in there.
Oh no, but they got to say that again. So when you see statements like this, the invariable conclusion I draw is literally, we have no idea what we're doing. Like, they have no idea what's going on. I don't think you can draw any other conclusion from a statement like that. The contradictions between the statements in box 2 and other things they've said in the paper. Again, this is a function of the fact that it's being written by committee. Every single person is familiar with some different niche. They have no organization whatsoever to like how gyms work on the immune activation of TLR4s. You know, it's all very like helter-skelter. And I said, haha, what does it say? The significant metabolic interrelationships of small dense LDL with abnormalities of other atherogenic apoB containing lipoproteins, particularly increased concentrations of VLDL and remnant lipoproteins, have created challenges in assessing the independent contributions of small dense LDL to CHD.
Well,
I was like, that's funny.
What do you know? Nevertheless.
And again, they start talking about, so they're like concentrations of small dense LDL, but not large LDL predicted, again, associated with incident CHD independent of LDL-C.
Okay.
Again,
what about remnants and stuff? They cancel those out.
But they said it's so that they're basically saying that LDL-C doesn't matter as long as you have large LDL. Like there was this paper I found, I don't know, it was a year ago or something, and it was like LDL protects against oxidized LDL. I'm like, so you're saying that non-oxidized LDL is good? Like that kind of thing comes up all the time where they're like, so they're saying basically that large dense LDL is totally fine because it doesn't predict operative word.
Oh, here's one that I like. Moreover, unsaturated cholesterol esters in the lipodome of small dense LDLs are markedly susceptible to hyperoxide formation under oxidative stress. So,
which could also be called metabolic syndrome.
Also, unsaturated fats.
Yeah.
Ending up in your unsaturated cholesterol.
So always remember to eat very saturated fats. Follow the croissant diet, everyone. It's the only way to health.
Just gotta load up on that stearic acid.
Anyways, we get into some mumbo jumbo. Let's see. So we've got responses elicited by low density lipoprotein retained in the arterial wall. Again, so they're trying to mentally draw the causal graph. I have a lot of question marks in this first bit here. The causal graph is that retention happens. They've admitted it happens after other things, and then the retention is causing something. So they say like retention and subsequent accumulation of LDL in the arterial wall triggers a number of events that initiate and propagate lesion development.
Okay.
Temporally, that's problematic because the accumulation of LDL, follows a lot of other structural changes in the artery wall.
Right. It's not just you get one in there and all of a sudden there's a pile of them and then bad things happen.
And again, I'm sure there's a feedback loop hypothesis somewhere in here. But they say due to the local microenvironment of the subendothelial matrix, LDL particles are susceptible to oxidation by both enzymatic and non-enzymatic mechanisms, which leads to generation of oxidized LDL containing,
But I like that they're just like, it's just the space. It's
due to the local microenvironment.
The local microenvironment's just oxidative-y, It's just...
It's
just taking off a lot of electrons. A lot of electrons being stolen there.
I think
dangerous part of town. Watch out.
Papers. It would be interesting to dig into a couple of these citations.
Hold on to those electrons.
I think a lot of times when you see citations like this, they will again not really support. Let's see, citation.
And then apparently our oxidized LDL creates a sterile inflammatory response by activating endothelial cells to upregulate adhesion molecules. And what do you bet those do? Probably cause LDL to stick. It's probably the wrong order of operations here.
Yeah, they get a lot of that causal arrow.
So what do we have for 129 and 130 in our citations?
129 is innate sensing of oxidation-specific epitopes in health and disease. Seems like an interesting thing to look at because it's again like that kind of idea that the immune system is associated with clearance of borked molecules. Like, that seems plausible. Let's see. Generation of biological activities of oxidized phospholipids.
It's not entirely clear to me that these support the point that the microenvironment leads to oxidation again for reasons.
Yeah.
But
anytime they say, like due to the science word of the whatever, like that, like...
with no cited source.
Yeah, whatever. And then... oxidized LDL in turn initiates a sterile... someone needs to actually draw the causal graph that they're arguing here. It's just like a haywire of non-connected.
They have a bunch of graphs in here, and they all kind of have a bunch of arrows and stuff, and they don't really
graphs with
point to much.
Things going up and down. Sterile inflammatory response. Did I pull 131? I forget if I did.
I don't think we looked at 131. No.
I printed two of these out. Atherosclerosis, current pathogenesis and therapeutic options. That doesn't seem like a great paper. At least, so they did say mice here. They said the importance of oxidized phospholipids in the inflammatory response of vascular wall has been demonstrated through the transgenic expression of an oxidized phospholipid neutralizing single chain antibody which protected atherosclerosis prone mice against lesion formation. Again, like, how relevant is the apoE mouse to human atherogenesis? A lot of these,
I'm going to go with not super relevant.
It seems to be probably relevant. The pathology has been reported to be similar to people with FH or like homozygous FH. But that differs somewhat markedly from people with without those sort of receptor mutations. So
and then I guess we start talking about macrophages and foam cells again.
Yeah, so they said macrophages exhibiting different phenotypes ranging from classical inflammatory subtypes to alternatively activated anti-inflammatory macrophages have been identified in atherosclerotic lesions.
And I've tweeted about this before. I found that the foam cells actually tend to be expressing anti-inflammatory genes, whereas the non-foamy cells express inflammatory genes. But I like that they're like, oh, the classical inflammatory type, you know?
Yeah. So
classic.
Let's see. And then it talks about a number of different things that could promote this. It goes over several DAMPs, generated by modification of retained LDL citation needed, induce
and what are these?
Induce the expression of pro-inflammatory and pro-thrombotic genes in macrophages by engaging in pattern recognition receptors such as toll-like receptors, which again, it's interesting that they're now bringing in the innate immune system, but they're hypothesizing that the innate immune system is driven by
OXLDL just triggers a handful of them. TLR2, TLR4, TLR5, TLR6.
Yeah.
And CD36 maybe, but I don't think that I've come across that OXLDL activates TLR4.
It's one of those things that, like, in a petri dish when you're just pouring it on top of a cell.
Yeah, you might get enough on there. The other interesting thing is you get other stuff that can act as TLR4 agonists and antagonists.
It says CD36 ligands promote sterile inflammation through the assembly of a toll-like receptor 4 and 6 heterodimer. Natural Immunology, 2010, Stewart.
Guess we should check that one out.
Might be interesting. Let me,
haven't read that one yet. Which citation was it? 139?
Yeah.
I can pull that up. Of course, they don't actually put.
So it's link.
Yeah, no links.
Yes. He always smells my mouth like. Have you had bacon recently?
In atherosclerosis and Alzheimer's disease, deposition of the altered self components, oxidized low-density lipoprotein and amyloid beta triggers a protracted sterile inflammatory response. Maybe.
Like, how do they
Although
know
chronic
that?
stimulation of the innate immune system is believed to underlie the pathology of the disease, the molecular mechanism of the activation is unclear. Here we show that oxidized LDL and amyloid beta trigger inflammatory signaling through the heterodimer of toll-like receptors 4 and 6.
I feel like they've made that critical assumption that's unsubstantiated that it is a sterile inflammatory response.
And I'm sure that this was done in mice or in a petri dish, even better.
I'm sure.
Results. Lipoproteins accumulate abnormally in the artery wall during hypercholesterolemia, setting off a cascade of pro-inflammatory events.
That is the initiating factor.
Particularly the expression of chemokines that initiate an influx of monocytes.
That entire hypothesis really bothers me because they talk, like, by any definition of reality, extensive discussions... Dog, don't chew on the table. Like, any... How to phrase this? Any discussion...
so CD36 is a TLR4, TLR6 co-receptor. So CD36 somehow
okay, dog, hold on. The dog is
drives expression of things via toll-like receptors. So the CD36 acts via TLR4, sort of. That's how I'm interpreting this.
But, like, what bothers me about these papers is they're like, so if we know for a fact that the accumulation of lipoproteins occurs later in the progression, then when they're, like, this entire discussion... They're oblivious to, like, they're not saying, you know, after a while, once the cholesterol accumulates, they're saying that, like, in the very beginning, in the dawn of time, LDL was deposited and then this happened. It means they're ignoring a large portion of the disease. And I don't know, I guess it depends how you characterize it. Do you characterize it as a long chronic disease? I know there are people who characterize it as a more acute disease, at least in the later stages. Moreover, SMCs may take up cholesterol-rich lipoproteins to become macrophage-like cells that contribute to the number of foam cells in advanced lesions. Interesting. So, and there's another citation needed quip here. They say, like, an important consequence of lipid loading of macrophages is the formation of cholesterol crystals, which activate intracellular complex, the NLRP3 inflammasome, to promote local production of IL-1 beta and IL-18. The 144 citation there, I think, is the one that most accurately supports that. I don't know why they cited the other two. I think they were vaguely tangentially related.
I could believe that a cholesterol crystal would cause a problem. But
I could believe
it's
that,
not
but
clear to me how, where, and under what concentrations and parameters these are actually formed.
It was a test tube experiment.
Are they found in all lesions? What type? You know, I feel like I don't have enough information here.
Yeah. This is why, like, pathology would be very interesting, because, like, pinning down exactly the list, the series, would be, I think, pretty important. They, like, again, 144 was in mice and in test tube. I think they had part of it that was both to make their overall point. But I like the question of how relevant, you know, pouring or quote unquote treating cultured cells with modified LDL in a test tube. Like, it's
a good starting point to rule out things, I guess.
Yeah.
You know,
I mean, I think it's probably more interesting of ruling out mechanisms than trying to argue like this is the key mechanism. But a lot of times they just kind of imply that it is. So then the next sentence is a citation needed sentence. The persistent presence of lipid-derived damps in the arterial wall, together with continuous expression of inflammatory cytokines and recruitment of phagocytes, whose role is to remove the triggers of inflammation, sustains the inflammatory response. And then there's another sentence. It also facilitates an active crosstalk with several other arterial cells, including mast cells, which in turn become activated and contribute to plaque progression by releasing specific mediators. The 145 citation is a somewhat interesting paper on mast cells, but it does not, like, that citation is not related to the previous sentence. There's just no...
Yeah, definitely need some more information there.
Oh, I found my pen. Let's see.
And then they're talking a lot about the adaptive immune system, which, to be honest, I really don't have a whole lot of knowledge about at this point. But they're talking about T helper cells of various types and the various cytokines and chemokines expressed by those.
Yeah.
Antibodies that they produce.
Call me an engineer, but I would love to first understand the extent to which the innate immune response to endotoxemia could be responsible and bound that, you know. If we can get some understanding of, like, well, through that pathway, like, what percentage of the progression can we explain? What percentage of the activation of the immune system, at least, can we explain?
Yeah, let's try not to throw in a whole nother immune system in there.
Because I know that there's a lot of interactions here. This is not entirely decoupled, you know. There are aspects of the adaptive immune system that will respond to these same stimuli, but let's see. Talk about some antibodies. Many of these antibodies have specificity for OXLDL and, in an isotope-dependent manner, trigger activation of complement, further modulating the immune response. The 152 citation, I think I printed it out. It was an apoE checkpoint inhibitor of inflammation. So it's talking about the... doesn't really talk a lot about apoB, but it was talking about... says, thus, the conclusion of the abstract is, thus apoE is a direct checkpoint inhibitor of unresolvable inflammation, and reducing C5 attenuates disease burden. C5.
No idea.
Treatment with siRNA against C5, which is formed by all complement pathways, attenuated murine CHP inflammation.
This seems like a very mechanistic paper that doesn't really support their...
Oh, no, I don't think it supports the hypothesis at all. I think it's kind of interesting to talk about, like, apoE and its association with different diseases.
Right.
I think it would be quite intriguing. We've kind of... I don't know if our point here is to talk about how all these things are interesting to our hypothesis, or how...
I think just talking about it.
Yeah, so
what comes of it.
So it says, thus, retention, it says, after a bunch of things that are, like, maybe tangentially related in some citations that supports them and some that are totally unrelated, they say, thus... retention and subsequent modification of LDL elicits both innate and adaptive cellular and humoral immune responses that drive inflammation in the arterial wall, no citation, and disrupting this vicious cycle by targeting inducers and mediators. Again,
they got a box, though.
What
Box 3.
Oh, we got box 3. What do we got in box 3?
Nothing good.
Sand in the gears hypothesis. So then we talk about defective efferocytosis, which I think is kind of an interesting discussion because that's one of the observations. Like, how interesting is... I'm curious to what extent... one of the observations that Velikan calls out is defective efferocytosis, a total absence of attempt at healing within the artery. To what extent is that what's going on, or to what extent is atherosclerosis actually a more acute disease? And to what it like? So if you talk about like a lot of the thrombotic hypothesis of progression might hold that it's actually a more acute idea. So, you know, perhaps it's progressive up until a certain point, and then once you get endothelial erosion, that'll start to precipitate this oscillating cycle of endothelial erosion, thrombus formation, reincorporation, you know, stabilization, but then, you know, further assault will cause it to... So you have this sort of... That could be a very acute progression model where, like, you know, it tries to stabilize it, but it ruptures again. And each progression is a very rapid event. You know, it ruptures, you have thrombus formation, you have reincorporation, and then
I think at that
slow
point
attempt
you would then have cells to efferocytose.
Oh, for sure.
So
and I think that,
you know, if you had your immune system was all bound up with doing something else because of endotoxemia or other issues, or if you were in a constant fed state and you had no whatchamacallit,
I'd be interested to see... I do want to understand the extent to which we could explain that aspect of the disease with the immune system being busy versus like hyperplasia and insufficient angiogenesis and just a starvation type mode. Because I know like hypoxia, like what does that do to the ability of our immune system to do efferocytosis? I also realized Shoban was explaining to me efferocytosis on Twitter, and like I haven't read up enough on like exactly how the immune system and efferocytosis are related. There's like clearly immune response and resolution of the response with healing. You know, there's this whole kind of cycle that's happening. So they have a...
They also talk about other things causing damps that further propagate inflammation. So that I don't know how they're ruling out, you know, the damps being created from necrosis and other sources of
I think
PAMPs potentially.
Generally, they kind of say something and then they consider that they've proved the point and then go on to talk about interesting things, assuming that they've proved the point that like LDL is the root cause of all evil.
Yep.
They did say this involves recognition of several eat-me signals, such as phosphatidylserine exposure, and moreover, don't eat me signals, such as CD47, also play a critical role. Don't eat me signals. Interesting. Let's see. Efficient efferocytosis protects against atherogenesis by removing cellular debris and creating an anti-inflammatory milieu. There are a number of things that are important to resolving inflammation.
Aptly called resolvins.
Resolvins. I do love the names. As a result, efferocytosis becomes defective and resolution of inflammation, which is mainly driven by modified LDL. Just in case we forgot that statement, no citation.
I missed that.
Is impaired. I love these things where you like circle the like, it's like Wikipedia citation needed. Under such conditions, apoptotic cells accumulate and undergo secondary necrosis, promoting the release of several damps that further propagate inflammation. I think that's totally true.
I've never heard of secondary necrosis before.
Me neither.
Didn't know cells
I
could
did
die
box
twice.
it with my pen. Thus, defective efferocytosis may be a potential therapeutic target.
I'd also note that there's no citation on secondary necrosis, which could just be totally made up.
You never know. Okay. Our knowledge of the intricate relationships between plaque stability and the cellular and non-cellular components of plaque tissue together with their spatial organization...
One more thing to go back to. Thus, defective efferocytosis may be a potential therapeutic target. We're throwing more sand in the gears. We got to...
Yeah.
All these things, we're just in search of a therapeutic target.
That's what we need. So they talk about how their knowledge of the intricate relationships between plaque stability and the cellular and non-cellular components of plaque tissue together with their spatial organization is incomplete. To me, when I was reading... what was the pathologist's name that we saw in the video?
I don't remember.
Okay, well, there was a pathologist who studied plaque rupture and had a great paper that I tweeted out 2 weeks ago. Stevens? It wasn't Stebbins. Stebbins was a different one. Davies, MJ Davies.
That was it, MJ Davies.
He had a great paper on rupture with pictures and everything from, I think, 2001 in Heart. And he seems to say rather definitively that plaque rupture and lack of stability is just a result of immune activation, basically. That they're essentially one in one. I don't know how they've determined that it's incomplete. I assume generally when they say like is incomplete, they're like, we haven't yet been able to tie it into OXLDL. Like that's kind of what they mean, which may be what they're getting at here. Local SMC, yeah. So they basically say it's incomplete, and then they start citing ADD style facts about OXLDL relation with these sorts of bits. That's kind of the thought process that I see when I.
Yeah, blah blah blah, necrotic core, blah blah blah.
I'm gonna just
cholesterol crystals again. Maybe we have those, maybe we don't.
Protruding through the plaque surface around the sites of rupture.
Intraplaque angiogenesis. That's pretty interesting.
Box 5.
Plaque rupture accounts for the majority of coronary thrombi at autopsy, apparently.
In men, not in women.
Yes.
Did they say that?
They did talk about that somewhere. Not in this section, apparently.
Or was that MJ Davies?
No, there was like a men versus women distinction somewhere in here.
Lesions without lipid cores or with thick fibrous caps are not at risk of rupture, but may produce a thrombus in response to plaque erosion. Do they mean endothelial erosion? Probably.
Possibly, yeah.
Again, vernacular in this field is a little touch and go. Underlying lesion is frequently but not always rich in the glycosaminoglycan hyaluronan and SMCs. Again, I think we've talked about the association between endotoxemia and vasospasm has also been proposed as the initiating event in plaque erosion. Interesting. Where did they get that from? That's an interesting citation. As you can tell,
oh, that is very interesting. Yes.
The mechanisms leading to intravascular thrombosis is not yet clear, but experiments with mouse are, oh, for fuck's sake. Subendothelial hyaluronan. Pelican talks about this, and I remember Googling it and showing that it was associated with endotoxemia. Most key terms that you can find in atherosclerosis are associated with endotoxemia.
Neutrophil-mediated denudation. Ooh.
Is that? Yeah, I mean, that makes sense. It's immune activation.
Mm-hmm.
It's all turtles all the way down.
Pretty much.
Erosion favors a higher fraction of thrombi in younger, especially female patients. So they did talk about that with very thin fibrous cap fibroatheromas.
Somewhere in here, I thought they actually talked about the hormonal component with estrogen.
Yeah, I mean, in general, I think this is a very interesting paper if you're not actually trying to understand how it supports the LDL is causal hypothesis. It's a bit ADD-ish. Like, in general, like, the major difference that I see when you're reading, like, Konstantin Velikin versus, like, a modern, like, paper on atherosclerosis is just the ADD nature of it. It's just like a word vomit of different random facts about LDL in no particular order that doesn't follow any, and they don't, like, unify things. They just kind of, like...
And, like, you can tell that they were trying to be clever with some of the, like, title section naming, like, fibrous cap matrix components, guardians of cardiovascular peace. Like, somebody got real happy, real excited about titling that sucker. They got, like, a whole paragraph, and
my God,
they loved it.
I didn't see that.
And the rest of them are just boring. Like, does aggressive low-density lipoprotein lowering positively impact the plaque? Like, that's just a good title section. I'm going to
guess that they're going to conclude that it does.
I think you're right. I think they do conclude that.
Doesn't matter that they have zero pathology to support that. They got some angiogram somewhere.
Like, some of the, like, they really do, like, they just jump in, like, thus, frequency-driven optical coherence tomography imaging of, like, did we need to know that? Like,
there's some really fun.
Couldn't you just say imaging? Gosh. Frequency-driven optical tomography, optical coherence tomography. That's fancy as shit.
There's one really funny quote where they were just like, atherosclerosis is a function of mechanical and other stimuli. And you're like, it's something like that, where you're like, are you kidding? Like, what? Like, there's no other possibility. Like, you haven't contributed anything to the...
There's clonal expansion of smooth muscle cells. They mentioned that.
Yeah, so let's see. Calcification.
I like that they mentioned the paradox of elite male endurance athletes with higher CAC scores than less physically active individuals, but with fewer cardiovascular events. So they have a paradox of whether calcified plaque influences rupture vulnerability either positively or negatively. Do they know? We do not know.
Well, I think in general it's known that the calcification, like calcified mature plaques, are not the ones that rupture.
Right. I mean, they're trying to like figure out like, is it that the minerals, or like we need to do finite element analysis of precisely how the crystal structure of the calcification, maybe running, like the impact of the running hardens the calcium. I don't know. I'm just
our imaging here is just so shit. Like, we just don't have really good hooks into the understanding.
I do like that they jumped to 18. What is this thing called? It's not labeled glucose. It's sodium, radioactive sodium fluoride, basically, with a PET CT. So they do positron emission tomography, where they give you a labeled sodium that's labeled with a isotope of fluorine, and then that emits positrons. And with a CT scan combined with a positron detector, you can detect where calcium is. So that was pretty cool that they were able to find really tiny.
Oh, interesting. But like
areas of stuff.
People can have a calcium score of 0 and have a heart attack.
But this one you can. But this one, the calcium, the labeled PET scan with the fluorine gives you a much better.
I understand it gives you a better resolution, but like, I think it's actually
able to. It's able to find lesions that are undergoing like active immune stuff. I looked into this once, and it was actually super interesting. I would love to have. I'm
curious, as a portion of lesions, what percentage of them would not be sensitive to this test?
I think we should look into it more, because it's probably one of the most promising tests. The problem is that it's a pretty large dose of radiation.
Well, that's fine. We're on a carnivorous diet. We can nuke our own digestive
all
systems.
those omega-3s are totally going to save me, according to the mice studies. If we
I really need to not eat omega-3s.
Statins and high-intensity exercise promote calcification without increased risk. Mineral morphology. Wow, they stabilize the mineral morphology. Do we need to phrase it that way?
Modified LDL stimulates vascular calcification by driving osteoblastic differentiation of vascular SMCs. That sounds tenuous.
No, I think that's... Oh, well, the differentiation part's true. I don't know about LDL.
I think the differentiation may drive calcification.
Yep.
But I don't know that the LDL... That's their, like, also their crux that the... That's the one of the... Because, so, like, if you understand that one of the initiating events is... de-differentiation of vascular smooth muscle cells, they'll try to show how like LDL does that. Mm-hmm. Although I think that especially because a lot of these de-differentiations occurring like long before substantial accumulation of LDL in the tissue. Yeah, it's one of the things that they if you actually, I forget which one of these papers I looked at, but they were talking about like, when the LDL concentration increases in the tissue. But you have to be very careful, because a lot of times they will conflate the tissue concentration of LDL with the serum concentration of LDL. And they're, sufficiently independent that you can have a blob of cholesterol in your tissue without having just like gobs of cholesterol circulating in your blood, just like floating in kind of large. Like the way that cholesterol gets under your intima is not because your blood turns white with the amount of like cholesterol accumulation. Right. Yeah, no, it's not.
Even they admitted this. So, can genes influence the susceptibility of the artery wall to coronary disease? I think we're just going to say yes. I don't really want to go over this section because I hate genetics and I don't like GWASs, and I think that they're kind of nonsensical.
In general, I think if we look at these, they'll all associate with immune.
Yeah, pretty much all of these. I mean, they talk about LDL receptor, CAV1, caveolin.
Which plaque components favor a thrombotic reaction upon rupture? That seems stupid.
So yeah, I mean, they look at LDL quality or quantity, PCSK9, HMG-CoA reductase, Niemann-Pick like 1, C1 like 1, the ATP binding cassette G5G8. So NPC1L1 and ABC G5G8 are both cholesterol absorption and cholesterol excretion. So that's like sort of interesting. You can get people who are hyperabsorbers and hyperexcretors and stuff. So that, genetics obviously play some role here, but I think
well, the problem
I haven't seen convincing GWAS, or not convincing GWAS data, but convincing data that this is actually clinically meaningful. That you can actually combine the scores and get a genome, a polygenic score that actually provides you with some kind of meaningful clinical outcome data.
Yeah.
If somebody knows of one, please send it to me.
I think part of the problem is that a lot of these studies are just hammered by the lipoprotein myopia to the point that they're not going to explore the extent to which these genes are doing other interesting immunological things that are more independent of LDL. They're kind of going to focus on that one pathway.
Well, yeah, that part, so they do a decent job of this because they look for all the genes that are associated with like having a heart attack or something. So the LDL ones show up, or maybe they include them or whatever. But you should, if you get enough of these things and they all have a big enough impact, you should be able to take someone's genome and say like, oh yeah, you have like all these genes, so you're going to die tomorrow, or you have none of these and you're going to be fine. But I don't think that these actually have enough of an impact to be clinically meaningful. Again, I think that the hazard ratios on a polygenic score comprised of like 300 genes is like 1.02.
That's hilarious.
I don't quote me on that, but I looked into this once and it was not super helpful.
Yeah. Interesting. So missing pieces of the puzzle. I love when they do these sections because they'll try their best to pretend that that they know. They'll give things that are somewhat orthogonal to the question at hand. Let's see. Yeah, so they'll do extension type things. They'll be like, genetic studies suggest that in addition to LDL, TG remnants and LPA are directly causal in ASCVD. What? Independent of LDL-C levels.
Oh, this
directly
is
causal.
This
so anytime
is MR,
they seem
this is MR studies.
So, yeah, they do talk
nonsensical.
about. I was like, but how do you? So they suggest that they are directly causal.
Yeah, if you jump down, they say using Mendelian randomization, blah blah blah. So just ignore any of that because Mendelian randomization is nonsense.
Can't show cause. Okay, the 3 lipoprotein classes may differ with respect to the mechanisms that underlie their respective contributions. I'm like, that's boring.
They talk about omega-3s.
Oh, that's good.
I think that probably omega-3s are good. They're from nature. Eat some fish. I don't know that you should be taking, ethyl isocosapentaethyl, which is just like super refined omega-3 pharma grade, but it did reduce C-reactive protein massively.
Interesting.
40% reduction in C-reactive protein. I mean, we're not really fucking around with that one.
I do wonder, what someone was mentioning that the Catavans or someone have surprisingly high CRP.
That's because of they have a parasitic infection almost always, and it's like a host, like they have a high basal inflammatory level, but it's like the parasite, it's hemolymph, something like that, some kind of wormy thing, and it excretes anti-inflammatory stuff, so it actually helps them. Weird. It's got a weird host parasite interaction thing going on. I looked into this once.
Vaguely commensalist. I'm curious. Yeah, I don't know. We need to our next step in all this lab equipment over here is to kind of try and pin down a good set of cytokines and things that we can test to try and evaluate this. And I think understanding the extent to which each of these cytokines is noisy, or there's genetic variation, or there's other confounders, is important to trying to narrow down which tests we're going to have LabCorp run.
We need to talk to them, yeah. So then, blah, 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 they mumble on about other stuff, and then they, again, state the unequivocal body of evidence for LDL causality in ASCVD will impact on future international recommendations for the management of atherogenic and ASCVD promoting dyslipidemias and will guide the rational use of both existing and new therapies. So it's all about therapies. And then they have finally this thesis has highlighted the emerging mechanistic features of atherosclerosis that can potentially lead to the evaluation of new therapeutic targets integral to arterial wall biology and plaque stability.
I just have a quick question. Are they admitting that they did all this stuff without any mechanistic evidence for 40 years?
Maybe.
This is a good question. I don't know. They're like, well, since we had no idea what we were doing then, because Subotin was pulling out these incredibly bold statements that were made that claimed that once statins were widely used, that heart disease would vanish off the face of the earth or something like that. And
guess what? Didn't work.
Surprise.
And then they finish with this diagram that makes absolutely no sense, with a bunch of arrows pointing at a bunch of other stuff that points at other arrows. It's
Ted Naiman did a knockoff of this with
an airplane.
Yeah, gravity is the cause of airplane crashes. It's not the cause. Oh, and then we get to the funding section.
Oh, yeah.
Ooh,
funding.
Boy.
Conflicts of interest.
Pfizer, Sanofi, Regency, Merck, Mendelium, non-financial support from Sigma Tau.
AstraZeneca, Pfizer, Sanofi, Regeneron, Merck.
And a personal honoraria for lectures and speakers bureau or consultancy from AstraZeneca. Genzyme,
Eli Lilly, Kowa, Amgen.
Pfizer, Sanofi.
Pretty much every drug company ever is mentioned here. We got a whole page dedicated to conflicts of interest. I didn't look up how much money these people got.
It doesn't say conflicts of interest. Oh, wait, it does say
it actually does, yeah.
I thought it interesting.
Anyway, they got a lot of conflicts of interest. So I think that, we didn't quite go through this whole thing because a lot of it was kind of boring and not really that interesting.
I think it was important for us ranting and raving will be helpful for other people to understand how we look at the paper and the reasons why we don't just see this paper and decide to choose a new career path and go and become vegan or something like that.
And if anybody has any questions about things we said or other things in the paper that you want us to talk about, or if we missed anything, critically interesting, please let us know. And if this was interesting to hear us rant about a paper for 30 or 40 minutes, we can pick another one and go at it again.
Do the other EAS paper.
Sure.
I don't know if I have time for that. We'll need multiple bottles of wine to get through that paper. Anyways, thank you for tuning in. Till next time.